Hello, I'm Patrick Wormley, and today I'll be examining the dermal absorption of titanium dioxide nanoparticles and the effect that it has on the dorsal root ganglia. Titanium dioxide is a metal oxide that can exist naturally as a salt, or it can be engineered into a variety of man-made nanomaterials. A nanomaterial is defined as any object with a dimension of less than 100 nanometers, with a nanoparticle referring specifically to a spherical one. Because they have such a small volume compared to the relative surface area, there's a lot more molecules on the surface of the object compared to the core, which changes the properties compared to a salt. As well as that, the surface of the nanoparticle can be functionalized with different compounds, which can also change the properties. As seen in the previous slide, the salt's white color is useful for many pigments, such as making white lines for tennis courts or coloring milk products. The nanoparticle, on the other hand, has many more useful things, such as sunscreen. It'll absorb UV light, which otherwise would cause sunburn and lead to skin cancer. As seen in this diagram here, the two circles and the giant U shape prevented the absorption of UV light from the skin, while the rest that was not covered had a sunburn. However, when you rub on sunblock, you're rubbing on a lot of nano titanium dioxide, which can result in dermal contact and potential for absorption. There are a lot of conflicting studies on whether nanoparticles cross the skin and are absorbed into the system. This is likely due to a lack of studies and a novelty of nanotoxicology. However, for this, we'll assume that titanium dioxide nanoparticles are absorbed slightly, and that this is also a result of long-term exposure of rubbing on sunscreen daily. The nanoparticles may enter through pores such as hair follicles and sweat glands, or it may travel between cells in the lamellar lipid bilayer. Zoom, zoom, zoom! The nanoparticle, with a log POW value of about 2.2, can pass through hydrophobic gaps and is also drug-like. The target of the nanoparticles is the dorsal root ganglia, which is a nodule of neurons that collect signals from sensory neurons and relay them through the spinal cord to the brain. Since neurons are very vital, they require oxygenation constantly, meaning there are plenty of arteries surrounding the ganglia. To get into the blood and enter circulation, titanium dioxide must first pass through the arterial walls. Following travel through the circulatory system and arriving at the target, it must cross back out through the arterial wall again and cross into the neuron membrane, which can occur either by phagocytosis or simple diffusion, depending on the functionalization of the nanoparticle. Once some titanium dioxide has crossed into the membrane, it can cause oxidative stress, such as lipid peroxidation, which affects the membrane, allowing more nanoparticles to leak through. Nanoparticles do not tend to be taken out of distribution, but rather just stay in the blood circulating throughout the system. However, some does get absorbed in the body, such as bone marrow and the liver. There's very few places where it can go without reacting. Because titanium dioxide nanoparticles are nothing more than inorganic compounds, there are not many reactions that can occur to the core itself. The compound is insoluble, so very little titanium is released as ions, which either way would not present any form or issue, as it is a vital metal to the body and has very little toxicity associated with it. There's 4 oxidation states of titanium, but titanium dioxide in the 4+ oxidation state is the only stable oxide within physiological conditions. So overall, for metabolism, titanium dioxide nanoparticles are extremely inert. Based on studies with oral consumption, there is little difference between the levels of titanium in urine and control the dose results, but there is a significant difference between that and the feces. This means that it does not pass through the gut lumen well, but there is also a size effect. Extremely small nanoparticles tend to be urinated, while larger ones tend to be defecated out. Due to its electrochemical properties, the parent dioxin produces superoxide, which is a result of an electron being passed from the surface to molecular oxygen, or quinones being attracted into the corona of the nanoparticle in redox cyclic reactions. Following its creation, superoxide can be converted to hydrogen peroxide via the enzyme superoxide dismutase, which can create hydroxyl radicals through Fenton chemistry. These reactive oxygen species can cause damage to nearly all cell components such as DNA, proteins, and any lipid membrane. As mentioned before, the target tissue is the dorsal root ganglia, which is a nodule of neurons that relay signals to the brain, which include the sense of touch, vibration, and spatial awareness. Inside the cell, one specific target molecule is DNA, which is contained within the nucleus. Titanium dioxide can cause DNA damage indirectly through the creation of oxidative species, such as the hydroxyl radical, but it can also directly affect the DNA by forming bonds with the backbone and between the bases. It can also affect other organelles and biomolecules throughout the cell, either by attracting into its corona, coordinating to the protein leading to a change in shape, or through the oxidative stress that it creates. The generation of superoxide leads to many different reactive oxygen, carbon, and nitrogen species, depending on the cell response. If this mutase is activated and peroxide is produced, then with the Fenton reaction, hydroxyl will be created. However, if it is not activated and nitric oxide radicals react with the superoxide, it'll form a larger nitrogenous compound, which, with the addition of carbon dioxide, can form two more radical species instead of having just one hydroxyl radical as a cell response would. However, either way, this is going to lead to cell damage. While it is not totally certain, there is some evidence that titanium dioxide nanoparticles may interact with DNA by inserting itself between base pairs or forming covalent bonds with the phosphate in the backbone. This could be a result of the coating of the nanoparticle, such as polyscyanine, or it could be from the core itself through titanium-oxygen phosphorus bonds from the backbone. In extreme cases, titanium dioxide can result in sensory ganglionitis, which is cell death or damage to the dorsal root ganglia. It can have mild symptoms such as numbness, but in more extreme cases can lead to serious problems such as motor skills and reflexes being compromised. While these symptoms may be serious in terms of daily life, they tend not to be terminal unless tumors are formed from genotoxicity. While titanium dioxide nanoparticles do have toxicity associated with them, they're also being tested for therapeutic uses. They can be used for anti-cancer drugs as both a delivering vehicle as well as a drug itself, which is just by changing the functional groups. One area of interest is binding antibodies to the nanoparticle, which will find a target and cause apoptosis to occur. This could be a huge development in brain tumors, which tend to be very difficult to treat. As well, it also has antimicrobial properties, which make it useful for disinfectants such as medical bandages and food processes. Nanotechnology is developing at an extremely quick rate and is expected to be a trillion-dollar industry within the next few years. However, its uses in the marketplace are being discovered faster than its potential dangers are. While there is some evidence that they may be a neurotoxin and carcinogen, it is proving to be a technology which may revolutionize not only medical science from treatment standpoint, but prevention even before diseases can start.